topic of this presentation is the use of spectroscopy in kinetics experiments. In this presentation, I will discuss the use of spectroscopy to experimentally determine the concentration of a colored species in a solution as a function of time for use in kinetic analysis. In the kinetics of acetone triiodide experiment, we used a Pasco spectrometer. We collected two types of data in this experiment. The first graph we generated was an absorbance versus wavelength graph. We then collected absorbance versus time graphs for 6 sets of initial concentrations. The following slides will explain the purpose and use of each of these graphs. We saw this slide in lecture about the use of spectroscopy and Beer's law to follow the concentration of a colored species in solution as a function of time. But now that we've done our first kinetics experiment in lab, let's review. In order for this technique to work, one of the reactants or products in the reaction must be colored. In this case, the triiodide ion, one of our reactants, has a yellow color. We placed a cuvette containing the solution into the sample holder of the spectrometer. A spectrometer works by shining light through the sample and comparing the intensity of light before it reaches the sample to the intensity of light after it passes through the sample. The instrument then calculates something called the absorbance, which is the negative log of the ratio of these two light intensities. In our experiment, after calibrating the spectrometer for dark and light, we measured the absorbance spectrum of our stock solution, which was 2.5 times 10 to the minus 3 molar of potassium triiodide. This plot shows how the yellow potassium triiodide solution absorbs light of different colors. The y-axis in this plot is the absorbance value measured by the spectrometer, and the x-axis is the wavelength of light. If we overlay the color spectrum onto this plot, we see that the potassium triiodide solution absorbs the strongest in the blue region of the spectrum. This means that the blue light is not reaching our eyes, which is why we observe the solution to have a yellow color. For this experiment, the experimenter chose to monitor the absorbance of light at 415.8 nanometers. The stock solution had an absorbance value of 1.310 at this particular wavelength. From the information in this plot and the known concentration of the stock solution, we can determine the Beer's law constant for potassium triiodide. Beer's law states that the absorbance of the solution will be directly proportional to the concentration of the absorbing species. This law holds true as long as you always measure the absorbance at the same wavelength of light. We used this simplified version of the Beer's law in our experiment. Since we know the absorbance of the solution, 1.310, and the concentration of the potassium triiodide, 2.5 times 10 to the minus 3 molar, we can then calculate a value for the Beer's law constant B. Some of you may have Googled Beer's law and found the following equation. Absorbance is equal to the squiggly E times C times L. In this version of Beer's law, the squiggly E is defined as the molar absorptivity of the solution at a given wavelength, and L is the path length of the sample, which is equal to the cuvette width. For our simplified version of the law, we combine the E and the L into one constant and call it B, where B is equal to E times L. We don't need to separately determine E or the path length of the cell, we can just use B. Next in the experiment, we use the spectrometer to monitor the absorbance of the reaction solution as a function of time at different initial concentrations of the reacting species. As the reaction proceeded, the absorbance of the solution decreased because the yellow triiodide ion was getting consumed. We generated 6 of these graphs in the experiment. 3 for the first set of concentrations, and 1 for each additional set of concentration values. By following the steps in the data analysis section of the lab manual for this experiment, you can convert the absorbance versus time graphs into concentration versus time graphs by using the value of the Beer's law constant obtained previously to convert all of the absorbances to concentrations. The Pasco software does this math for you. You just have to put in the value of B. Then you will use the line fit function in the software to determine the initial slope from each plot. Recall from lecture that the initial slope is proportional to the initial rate. Starting with the 6 absorbance versus time graphs, you'll use your Beer's law constant to get 6 concentration versus time graphs. From these, you will find the initial slope to determine the initial rate for each run. To complete the analysis of the data, you will use the method of initial rates to determine the rate law and rate constant for this reaction.